Good afternoon, sir. Myself Akshay Shivdas Gazbare, MSc first year biotechnology student. Now I'm going to express seminar on morphology of bacteria, and this is the subject of general microbiology. What is bacteria? Bacteria is unicellular, free-living microscopic microorganism which is capable of performing all the essential functions of life. Bacteria contain deoxyribonucleic acid and ribonucleic acid. Bacteria are prokaryotic microorganisms that do not contain chlorophyll. They are mostly found in the water, soil, air, food, and all the natural environment. They can survive in extreme conditions like in high temperature, in high pH, oxygen level, and atmospheric pressure. Size of bacteria. Bacteria are very small microorganisms which are visible under the microscope. They are having the size range in microns. Bacteria are stained by staining reagents and then visualized under high power of magnification, about 1000x of compound microscope. An electron microscope is used for clear visualization of internal structure of bacteria. Shape of bacteria. On the basis of shape, bacteria are classified as follows. We are seeing here the classifications of bacteria. First is cocci, bacilli, vibrio, spirulia, spirochetes, actinomyces, and mycoplasms. This is the image of cocci bacteria. Cocci are very small, spherical or oval shaped cells. In Greek, coccus means berry. It looks like berries, and the example of cocci bacteria is Micrococcus bacilli. They are rod shaped cells. Examples of bacilli are Bacillus anthracis. It is derived from Greek word bacilli, means stick. It looks like a stick. In some of the bacilli, the length of the cell may be equal to the weight. Such bacillary forms are known as cocobacilli. Vibro. This type of bacteria is like a comma shaped or curved rod. And the example is the vibro comma spiralia. Spiralia are the bacteria which has uh, long length and which looks like a spring. They have a helical shape and rigid body. An example of spiralia is Spiralium reprium, spirochetes. They are slender and flexuous spiral forms. A general structure of bacteria. In a bacterial cell, bacteria contain flagella, pili, capsule, plasma membrane, cytoplasm, cell wall, ribosomes, mesosomes, inclusions, and spores. In this picture, we are seeing a long part which helps bacteria for the movement, that is flagella, and also cilia may be present on cell, but here we are saying pili also helps in the movement of bacteria. Flagella are long, slender, thin hair-like structure, cytoplasmic appendage which are responsible for the motility of bacteria. These are the organ of locomotion. They are 0.1 to 0.2 micron in diameter, 3 to 20 micron in length. Flagella are made up of the protein flagellin. The flagellum has three basic parts. First is filament, hook, and basal body. These three parts composed in the formation of flagella. Filament is a thin, cylindrical, long outermost region with a constant diameter. The filament is attached to a slightly wider hook. The basal body of composed of small central rod inserted into the series of rings. Gram negative bacteria contain four rings as L ring, P ring, S ring, M ring, where gram positive have only S and M ring in basal body. Pili or fibrae. Pili are hair-like microfibrils which has a length about 0.5 to 2 micron in meter and 5 to 7 nanometer in diameter. They are thinner, shorter and more numerous than flagella. They are present only on gram-negative cells. They are composed of protein known as pillin. Pillin. They are unrelated to motility and are found on motile and non-motile cells. Fibrae and pili, these are two terms are used interchangeable but they can be distinguished. Fibrae can be evenly distributed over the entire surface of the cell or they occur at the poles of the bacterial cell. Each bacteria possesses 100 to 200 fibrae. Pili are usually longer than fibrae and number of 1 or 2 per cells. Function. Pili play an important role in attachment of the surface. Hence, pili is also called organ of adhesions. Plasma membrane. The cytoplasmic plasma membrane is a thin and has 5 to 10 nanometer in size. It separates the cell wall and cytoplasm. It composed of phospholipids and proteins. Prokaryotic plasma membrane are less rigid than eukaryotic membrane due to lack of sterols. Functions of plasma membrane. It acts as a semi-permeable membrane controlling the inflow and outflow of metabolites to the form and protoplasm. It provides the mechanical strain to the bacterial cell. It helps in DNA replication. It contains enzyme primase which plays an important role in the passage of selective nutrients through the membrane. Cytoplasm. The bacterial cytoplasm is a suspension of organic inorganic solutes in a viscous water solution. The cytoplasm of bacteria differ from that of the higher eukaryotic microorganism in not containing cytoplasmic reticulum, Golgi apparatus, mitochondria and lysosomes. It contains the ribosomes, proteins and other water soluble component and reserve material. In most bacteria, uh, extra chromosomal DNA called plasmid DNA is also present. Mesosomes. Mesosomes are respiratory site of bacteria. The mesosomes are attached to the bacterial chromosome and involved in DNA segregation during cell division. They are predominant in gram-positive bacteria. Nucleoid. The bacterial chromosome is not surrounded by nuclear membrane, so it is called a nucleoid. The bacterial chromosome are made up of double-stranded circular DNA. Intracytoplasmic inclusion. Many species of bacteria produces cytoplasmic inclusion bodies which appear as round granules. They are made up of either glycogen or starch. They appear reddish when stained with polychrome methyl blue or toluidine blue. Capsule. Bacteria synthesize loose amorphous organic exopolymer which is deposited outside and tightly to the cell wall called a capsule. Capsules may be composed of complex polypeptide or polysaccharide. It has 98% of water, which is the main component of the bacterial capsule. Sometimes the capsular material is loosely associated with the bacterium. It can be easily washed away. The loss layer is called a slime layer. Capsulated bacteria produces smooth colonies and non-capsulated bacteria produces rough colonies on the surface of agar media. Function. They protect the cell from drying. They protect the bacterial cell against the antibacterial agents and phages. Cell wall. Cell wall is rigid structure which gives definite shape to the cell situated between the capsule and cytoplasmic membrane. It is about 10 to 20 nanometer in thickness and constitute 20 to 30% of dry weight of cell. The cell wall cannot be seen by direct light microscope and does not stain easily by different staining reagents. The cell wall of bacteria contain diamino uh, pymalic acid, muriatic acid, and thioic acid. These substrates are joined together to give rise to complex polymeric structure known as polypeptidoglycan or murine. Uh, Peptidoglycan is the major constituent of the cell wall of gram-positive bacteria, whereas in gram-negative bacteria, cell wall is present is only 10 to 5%. Functions of cell wall. Cell wall is involved in the growth and cell division of bacteria. It gives shape to the cell wall. It gives protection to internal structure and acts as the supporting layer to prevent rupture of bacteria caused osmotic pressure, different differences between intracellular and extracellular environment, to provide support for flagella, to regulate the certain degree of passage of molecules in and out of the cell. It serves as the site of attachment for most bacterial viruses. Ribosomes. Ribosomes are the center of protein synthesis in bacteria. They are slightly smaller than eukaryotic ribosomes. The sedimentation constant is 17 S subunit. These 17 S ribosomes are made up of two subunits, namely larger 50S and smaller subunit is 30S. During active protein synthesis, the ribosomes are associated with mRNA, and such associations are called polysomes. Spores. The process of endospore formation is known as sporulation, and it may take 4 to 8 hours in vegetative cell. Endospore are thick-walled, highly refractile bodies that are produced one per cell. Each bacterial spore on germination form a single vegetative cell. Therefore, sporulation in bacteria is a method of preserving and not reproduction. Spores are extremely resistant to the dissociation stainings, disinfectants, chemicals, radiation, and heat. They remain viable for centuries and helps bacteria to survive for long period under unfavorable condition. Endospore can remain dominant for thousands of years. Spores of all medically important bacteria are destroyed by moist heat sterilization, like autoclave at 121 degrees Celsius temperature for 20 minutes. Thank you.